Hello, everyone. My name is Jacob Martin. I am a master's student in the physics department at the University of South Carolina, and I do research for the mechanical engineering department. Today, I will be presenting my paper on the measurement of magnetic particle concentrations in wildfire ash via compact NMR. So first off, why do we want to monitor the magnetic contents of wildfire ash? So when a wildfire occurs, the ash can be deposited in a couple different ways. Firstly, it can be deposited directly onto the topsoil, as shown here. And secondly, it can be deposited through runoff water when there's a rainfall event following the wildfire that pretty much drains the ashes into nearby rivers, lakes, or other nearby bodies of water. And specifically, within those ash deposits, there are a lot of different iron oxides. And even more specifically, within the iron oxides, there are magnetic iron. And we're going to be looking at magnetite and magamite specifically. And for topsoil, when the ash deposits are deposited in the topsoil, the magnetic contents of topsoil are very closely related to climate and rainfall conditions for that environment, and the magnetic content of the soil can be used as a proxy parameter for determining those things in the environment. And for runoff water, we know that nanoscale magnetite has been linked to brain disease such as Alzheimer's. So being able to understand the reaches of the deposition of the magnetic contents of the ash can be very important for understanding the fire severity as well as the reaches of this deposition. So the goals here are to be able to quickly assess the total magnetic particle concentrations in ash and water, that being magnetite and magamite, and the public health schools we worked with on this, and they were currently using a synchrotron to analyze the ash samples, which was quite complex and time consuming and also not portable for them. And we were really wanting to be able to develop a system for in situ monitoring of magnetic contents in nearby bodies of water. So we found that the best candidate would be nuclear magnetic resonance. So a brief overview of nuclear magnetic resonance techniques. So the main one, or the most primary one that's used nowadays, is high field NMR spectroscopy, which is where you pulse the nuclei that you're looking at with a magnetic field in the megahertz range, or usually megahertz to gigahertz range. And then you collect this impulse response here, and you can analyze it as a function of frequency to get a lot of data about the molecular structures of the nuclei that you're analyzing. And you can also analyze the decay rate, which is how fast it decays over time, to understand the environment surrounding those nuclei, because the rate at which they decay can tell you about how quickly they're giving energy back to their environment, so you can know things about what is surrounding them. But that, on the downside, is very costly. It's not portable because the magnets that you need for this are huge and usually cryogenically cooled or use superconductors, and it's very difficult to achieve the homogeneity and the strength needed for these type of measurements. But for low field NMR relaxometry, you do get low quality molecular structure data, but the benefit is that you can still get information about the environment around the nucleus, because for low field NMR relaxometry, even though you have a pretty low inhomogeneous field, you can still get a response from the sample and then repulse that sample to keep getting these continuous echoes and then build up that decay curve over time to again understand the environment surrounding the nuclei that you're actually pulsing with energy. So for specifically NMR relaxometry with magnetic particles, it's pretty well known that transverse relaxation rates for magnetic particles within water are very linearly correlated to the amount of magnetic particles in them, or the magnetic particle concentration. And this is just an example of that linear regime here in a paper by Gunn. And so what we wanted to do is dissolve the ashes or wildfire ashes in water, and then use NMR relaxometry to measure the decay rates to get a quick understanding or a quick quantitative measurement of the amount of magnetic contents in that wildfire ash, and also in the water itself, or if we just had a sample of water. So the first component to creating this system is the permanent magnet array, and it is composed of N42 neodymium magnets, arranged as shown here on the right. We have two these large magnets here provide the bulk of the field strength, with these smaller ones acting as return paths to provide extra strength to the field and also further focus it around the yolks, and these steel yolks act as return paths for the flux, as you can see here in the simulation. And we also added steel caps to further improve the homogeneity of the magnetic field within this gap. And we did a 2D finite element simulation, as you can see here, which estimated the field strength to be around 0.65 Tesla, which is usually overestimates the field strength, and as you'll see it did here. But again, this is where the sample would go right here in the middle. So actually assembling the magnet, we used these two different types of magnets here. These bigger block ones were placed in the bottom and top of this 3D printed guide, and the rectangular ones were placed on the sides here. And then you can see the steel bars or the steel yokes are placed around the outside. And this is all assembled by hand. It's all held together purely by magnetic forces. I can't get it apart now that it's together. And it weighs about 4.5 pounds roughly. We measured a field strength of 0.565 Tesla at room temperature, and also found that it had a gradient of about a negative 800 ppm per Kelvin. So as the temperature increases, the strength will decrease a little bit, and we have to be able to account for that because the operating frequency is purely based on the magnetic field. This value here is just the gyromagnetic ratio of hydrogen nuclei. So at room temperature, it operates right around 24 megahertz. So for the signal processing and amplification electronics, you can see the full diagram here. We matched all the PCB traces and all the ports to 50 ohms to reduce losses throughout the system. And so pretty much what happens is we generate that 24 megahertz sine wave out of here. It goes through a power splitter to a mixer and a switch, which is then, the switch is then toggled by a pulse generator to create those RF pulses that go into the sample down here. They're amplified to about 35 dBm before going into the probe, which is isolated from the low noise amplifier by this pi filter here. And once the sample is excited, the response travels out of here. It's usually in the microvolt level. It travels through one low noise amplifier, which amplifies it by 40 dBs. It's mixed down to about the audio frequency range using the original sinusoid that was used to excite it, and then amplified again by another 40 dBs, filtered, and then sent to the oscilloscope to be acquired. So it goes through about 80 dBs of total amplification. So for the signal generation, control, and the data acquisition portion, we have an NI-PXI chassis that's fitted with an arbitrary waveform generator that generates the 24 megahertz sine wave we need, and then the pulse train generator, which generates a sequence of pulses known as a CPMG, or car per cell Meissen-Gill pulse sequence. This is what we use to pulse that switch to create the RF pulses, 24 megahertz sine wave. And our 90-degree pulse is about 6 microseconds, roughly. And that's purely just based on the amount of power that we're amplifying the signal to initially. And the tau that we use is 0.625 milliseconds, which you can see here. That's just the separation between the successive pulses of RF energy. So the GUI here, it can be seen on the right. Going back to the magnetic field gradient with temperature, since we have to take that into effect, we use a thermocouple to measure the temperature surrounding the magnet. And then using that, it's a pretty linear regime of how it decreases with temperature. We can calibrate to the optimal operating frequency to run the sample at. And so here at the GUI, you can see this is, you can run it for as many scans as you want. We usually do about 8, which takes roughly 2 minutes. But you can see some of the first few echoes here of a sample. You have your 90-degree pulse, tau, 180-degree pulse, and then an echo. And we pretty much take the peaks of all these echoes, usually thousands of them, and use them to create this decaying signal here shown in the top left and bottom right. And the transverse decay model shown here is what we use to model these functions and to extract a single decay rate value. So this shows the full desktop system. You can see the GUI here on the left with the NI-PXI chassis. Here are all the signal routing and amplification electronics. We have some DC voltage regulators that step 24 volts down to 12, 5, and 1.8. And you can see the magnet, the permanent magnet array, as well as the samples. So it's a fairly compact system. fits on the top of my head. So we took 10 total ash samples. These are samples that had already been previously analyzed using the synchrotron by the School of Public Health. So they had a pretty good idea of all the contents within them. We took 20 milligrams of each one and with 20 milliliters of water and sonicated them to make sure that everything was evenly distributed. Distilled water was taken as a reference for water without anything in it, any magnetic particle concentrations. And you can see some of them shown here. These are just 3 of the samples of different colors, along with distilled water, with distilled water, of course, having the slowest relaxation because it doesn't have any magnetic content. And we extracted the relaxation rates of all these using least squared or the least squares regression, again, with this model shown here. So when we plotted the R2 rates versus the concentration determined via synchrotron analysis, we did confirm that the relationship, the linear relationship expected was there. So we, and this was a combination of magmite and magnetite together. They measured it initially in milligrams per gram, and then we converted that to milligrams per milliliter unit, just using the amount of sample and the amount of water that we used. So again, this linear relationship is verified, and it was a pretty good fit. So this showed that we could use this method as a quick way to determine, the general amount of magnetic content within ash or within a sample of water. So, coming to a close, again, we showed that compact NMR could be used as a viable method to estimate or quantitatively determine magnetic content or magnetic particle concentrations within wildfire ash and water. And the robust and the compact permanent magnet array that I showed was sufficient for 5 millimeter NMR tubes, which is the standard size for NMR tubes. And again, it's a simple and rapid test compared to what they were already doing. You just pretty much mix the sample with water, sonicate it, and then it's ready to go. And we also, like I was saying, we're working to implement this in situ and reducing the size of the system so this can be set up at an inlet to a body of water or just at any type of body of water so that we can monitor magnetic concentrations or magnetic particles present in different areas over the course of days, weeks, however long it might be, or if it's just after a wildfire event, a rainfall event, anything like that. Thank you. That's all. Any questions?